In this lecture, we'll cover some biotechnologies. Biotechnology in general is studying, analyzing, understanding, and using genetic information. Since biotechnology involves utilizing genetic information, then we're obviously going to be talking about techniques that deal with DNA. And this here is sort of an outline of what the various techniques that we're going to talk about can do with DNA. So the first type of technique that we'll discuss the goal is to sequence DNA. And there's two techniques that I would normally talk about. They are dideoxy sequencing or Sanger sequencing and next generation sequencing. Since we've already covered dideoxy sequencing, I'm not going to cover it in here. Please refer to your lab notes for that technique. There will be questions about dideoxy sequencing on the final exam. So we'll focus on next generation sequencing. Now, in brief, why you might want to get the sequence of the DNA. It could be for identity, like with your fish DNA barcoding experiment. You wanted to confirm the identity of the species. Perhaps you're just simply wanting to build a restriction site map. You want to know where in this sequence are there sites for various restriction endonucleases where they would cut. Or maybe you're doing comparative studies, you're looking at sequences of different species and you want to compare how similar or different they are for evolutionary studies. Or maybe you are doing a diagnostic test, so you're trying to determine if somebody has a particular mutation, for example, and so you need to know the sequence of the DNA. But of course, there are many other applications. Since we did already talk about dideoxy sequencing, I'm just going to have these slides here just so that they're all together and to trigger your memory. But please, again, refer to your notes for lab. A newer and alternative method to Sanger sequencing is called next generation sequencing, or it's also known as high throughput sequencing. This term actually describes like 2 or 3 different types of sequencing, and we're going to talk about one of them. But really quickly, the big benefit of this method over Sanger sequencing is that you can sequence a lot more DNA at the same time. And so, for example, you could take a whole genome from a cell, like you see in this picture up at the top left. So you could take the genomic DNA, remove it from the cell, and first what you'd have to do, you see number 1 here, is cut all of that DNA into much smaller pieces. And these pieces can be up to 1,000 base pairs long, or 1 kilobase. And I'm going to be very brief about the next part. But what is then done is they take each individual piece and they'll, as you see here, they put it in with one little droplet of liquid or solution and a bead. Then they PCR or copy up that piece of DNA, and at the same time that they're doing that, they also add special ends to the piece of DNA and all of its copies, so that one of the strands actually gets attached to the bead itself. And so that's what you're seeing here. So what you ultimately end up with is a bead in a droplet with many copies of the same piece of DNA attached to the bead. And this is going to happen for every single piece of DNA that you want to sequence. Each piece will be in its own droplet with its own bead and the many copies. Each bead is then placed into a well, and like you see here, you can have this plate with multiple wells. Each well represents a different piece from the original larger piece of DNA that you cut up. And so if each piece represents 1,000 base pairs and each well has a different piece, imagine all of the sequence that you can get in one, what they call one read or one experiment, one run of this DNA sequencing reaction. So each bead with the many copies of one piece of DNA is placed in a well. And in that well is also placed DNA polymerase, right? Because you want to do some copying, or we're going to do a sequencing reaction. Primers, because as we know, DNA polymerase cannot start synthesis on its own. And of course you would have buffer. Now, usually we would also add DNTPs, but we're not going to add that yet. So right now we essentially have everything we need except for the DNTPs. Now that's going to be the trick to this reaction. What's going to happen next? So you have all the sequences ready to go, right, with their primer and with the DNA polymerase. It's just waiting for the DNTPs. So everybody's ready to go. What you're going to do next is to all the wells at the same time, we're going to add only one DNTP. And so we're going to look down here. So let's say the first DNTP that's going to be added is the nucleotide containing the adenine base. So all the wells are going to get that at the same time, just that one nucleotide. But not every single sequence that's in each of the wells is going to need an A to insert first, or a nucleotide containing the A base to add first. Only the sequences that have a T on the template next will need a nucleotide containing A first. And so you see that down here in the example. So this is supposed to be a bead right here. So this is one of the wells. This is supposed to be a bead. Here's the DNA template or sequence that we want to sequence. And it's attached to the bead. Here's the primer. And so DNA polymerase in pink here is ready to go. And you can see that the next letter after the primer is a T on the template. So the next nucleotide that should be added should contain an A. So as we add that nucleotide, it will get incorporated into the strand, into the growing strand. If that happens, if you guys remember, nucleotides, when they're free, have 3 phosphates attached to them. So when they actually get incorporated into a growing DNA strand during this sequencing reaction, 2 of the phosphates will have to be removed, and that's what you see right here. Those 2 phosphates or inorganic phosphates, they leave as a molecule called pyrophosphate. And you can see that happening right down here. Okay, so this got incorporated, you're getting a pyrophosphate. So if this happens in any of these sequences that we're trying to read inside of all the different wells, so let's say it happened here, it happened here, it happened here, it happened here, it happened here. So wherever that nucleotide containing an A got incorporated, like it did in this example down here, then actually a light signal will get released. I'm skipping some details about how that that little light signal gets released, but that's what's going to happen. It's going to get released as a result of the pyrophosphate being released. And so there will be a little flash of light wherever the A nucleotide was incorporated. And a computer will be able to record in which wells you saw a flash of light. And all the wells that did not release a flash of light, what that means is the A base was not the next in the sequence. That's not what is the next nucleotide in that particular template sequence. So once the computer records that, then all the unincorporated nucleotide containing the A base gets washed away. And then we flood the slide here, or all the wells, we flood the wells with the next nucleotide. And this time it's going to be a nucleotide containing thymine. And the same thing, the same idea. If thymine is next, if that's the right nucleotide in any of the sequences, then we'll see a flash of light, right? In those wells where thymine was incorporated. We'll see that flash of light because a pyrophosphate was released. Now in our example down below, in this particular well, in the template sequence, the next nucleotide that should be added is not one containing a thymine. So that does not get incorporated, no flash of light. And that's it. Nothing will happen in that well, so we don't see anything. The computer won't record anything. It won't record that thymine was the next base in the sequence. So then again, we wash out any unincorporated nucleotide containing thymine, and then we move on to the next nucleotide. This time it's the one containing guanine. We flood all the wells with nucleotide containing guanine to see if it gets incorporated. So we look for the flashes of light again to see if it was. In our example down here in this well, when it got the nucleotide containing guanine, it did not get incorporated, so no pyrophosphate, because this is not the next one that should be incorporated there. So this particular well would not flash a color or flash a light. Then again, we remove all the nucleotide containing, any unbound nucleotide containing guanine, and then we flood all of the wells with the next nucleotide containing cytosine. In this case, in our example well, the next nucleotide should be one that does contain cytosine, so it will be incorporated, we will get pyrophosphate released, and we will get a flash that the computer will record for that particular well. So the computer will know, or the computer will record that the next base in the sequence should be a C. And this would be one full round of nucleotide additions. Once this round is done and we wash out any unincorporated nucleotide containing cytosine, we'll repeat the whole rounds again. We'll go with another round and we'll keep doing the same thing over and over and over and recording who flashes during what nucleotide. This will happen in many, many rounds until you no longer get any more flashes from any wells after a full round. If that happens, then that means no sequences are incorporating any nucleotides anymore and sequencing is done. Sometimes students ask, well, what if there are two of the same nucleotide that would be added? There's two in a row. And so in that case, what would happen is when it's time for that nucleotide, when it's actually being added and it's next to be incorporated, like you see here, the next two bases or nucleotides that should be added should contain a thymine. So when the nucleotide containing thymine is added, two will be incorporated and you'll get an even brighter, more intense flash because more pyrophosphate will be released. And the computer will be able to detect that it was a greater flash indicating more than one nucleotide was incorporated. Down below, this is just giving you an example of the multiple rounds that might occur to kind of just give you a flow of a whole round of nucleotides being added one at a time. So here's our polymerase in purple, here's the template sequence attached to the bead, this is the primer, and we see that when thymine was flooded into the wells, a couple got incorporated because there must be two adenines in the template, and so two nucleotides containing thymine were added, and you got a flash of light from the pyrophosphate. Then when adenine was added or flooded to the wells, apparently that was not the next one, so it wasn't incorporated, nothing happened, no flash of light. Then guanine was added to the wells. It looks like that one is needed next, and so DNA polymerase incorporated it, like you see here, and you got a flash of light because of pyrophosphate being released. Then the next base to be added, or nucleotide to be added, is a nucleotide with cytosine base. It looks like that was not the next one that should be added, so nothing happened, no flash of light in this case. And so the computer will not record that cytosine was next. Instead, we have to now go back to the beginning and start with the next round of nucleotide additions. And that, in a nutshell, is next generation sequencing. The next category of techniques would be when you want to amplify or make many copies of a piece of DNA. There are two types of techniques that I would normally talk about. We've already extensively discussed one, that's PCR, so rely on your notes from lab for PCR. There will be questions that could deal with PCR in the lab and the lecture final, So instead, I'm going to focus on an alternative technique to amplify DNA called DNA cloning.
We did somewhat talk about this already. I'm just going to add a few details.
As you can see on this slide, DNA cloning, so the goal is to make many copies of a piece of DNA, but instead of doing a reaction in a tube, what you're going to do is you're going to basically use an organism, a living organism, and it's usually something like bacteria or a virus or yeast, but you're going to use an organism to make copies for you. You're going to use that organism's process of DNA replication. You're just going to trick the organism into thinking it's copying its own DNA. The example that I'm going to show you is what we would do when we are trying to use bacteria to make many copies of a piece of DNA for us. And in a nutshell, what's involved with this process is you would first have to isolate your gene or DNA piece that you want to copy. You would isolate it, and then you would do what we would call, you would insert that piece of DNA into a cloning vector. And so that's what you're seeing here. In blue is the piece of DNA that we want to copy, and we're going to insert it into another piece of DNA that's going to carry our piece of interest and allow us to put it into a bacteria cell. Now, the trick is to get that piece of DNA into your specific cloning vector. Now, before I talk about that, I just want to say a few things about different cloning vectors. And down below, here's the official definition that we're going to use of a vector. It's typically a DNA molecule, and again, it's just a vehicle to carry your genetic material of interest into another cell where it can be replicated or even used or even like turned on, expressed.
And what would be needed to accomplish this would be restriction endonucleases and ligase, which we have talked about before.
Here is a list of some commonly used vectors, and they're split up based on whether or not we're trying to put our DNA of interest into a bacteria cell or if we're trying to put it into a eukaryotic cell. We will focus more on plasmids, but before I do that, I'll just mention what the other ones are. So some other vectors for putting a piece of DNA into a bacteria cell are, for example, phage vectors. Phage is basically another term for a bacterial virus. So sometimes scientists use a virus that infects bacteria to carry their DNA of interest because the viruses have easy way of getting DNA in to bacteria cells. Or there are large pieces of DNA called bacterial artificial chromosomes, or BACs for short, and these also can be used to carry your DNA of interest. How these different vectors, how they differ from each other is mainly how large of a piece of DNA can you shove into it, right? Remember, these are going to be carrying your DNA of interest. And so in parentheses, what I've written down is pretty much like the upper limit of size of a piece of DNA that you can put into each of these different vectors. So if you have a really large piece of DNA that you want to get copied, you wouldn't be able to put it into a plasmid. It's more appropriate that you put it into something like an artificial chromosome. For eukaryotic cells, we also can often use plasmids, or we can use what we call YACs for short, yeast artificial chromosomes. And again, these are appropriate for holding larger pieces of DNA.
So again, we have to choose the right cloning vector depending on what organism we plan to do the copying in. And what we also have to recognize about a cloning vector is that it must have all the necessary sequences to get copied inside of, say, a bacteria cell, if that's where we're placing it into. So we need an origin of replication, for example. Or if we're placing it into a eukaryotic cell, we need the appropriate origin sequences there as well. We need to make sure that whatever vector we use is going to get copied.
So on this slide, it's just showing you what is characteristically seen in plasmids that are used for cloning, DNA cloning. And we've talked a lot about this before in lab. but if you remember, a plasmid is a circular double-stranded piece of DNA. They do normally occur in bacteria, but they have been extensively re-engineered for purposes in the lab, for use in the laboratory. And so typically what we find on a plasmid that we use in lab are these three things that are circled. An ORI, or origin of replication, so you see that right here. What the origin of replication does is it ensures that whenever the bacteria cell starts replicating its own chromosome, it will also start replicating this little plasmid piece of DNA as well. A second thing that is often seen in plasmids is a selectable marker. If you remember, this is a gene that will allow us to select the cells that got the plasmid and not allow the cells that didn't get the plasmid, not allow them to grow. It allows us to only select the cells that actually have our plasmid of interest. And in lab, the selectable marker we used was the ampicillin resistance gene. The third thing that we did not talk about in lab is often plasmids for cloning a piece of DNA contain what we call a polycloning site, or it's also known as a multiple cloning site, or MCS. And all that refers to is an area of the plasmid where scientists have engineered a ton of unique restriction sites where various restriction endonucleases would recognize their sequence and can cut there if you incubate this plasmid DNA with the respective restriction endonuclease. So as you see on this picture right here, it has a variety of restriction endonuclease names and little arrows pointing. That's telling you that, for example, there is a sequence here that the restriction endonuclease HIND3 would recognize and would cut, or a site that EcoR1 would recognize and cut, or SAL1 would recognize and cut, or BAMH1, etc. Now, the reason why this is there, this MCS or multiple cloning site is present, is remember that we're going to use this plasmid as a vector to carry our DNA of interest. We're going to have to insert our piece of DNA into this, which means we're going to have to cut it and paste our piece of DNA inside. So to give us more flexibility of where to cut and how to cut, that's why we have all these various restriction sites that we can choose from to cut open our plasmid DNA and insert our DNA of interest. Aside from a plasmid, we're also going to need restriction endonucleases because that's how we're going to cut our plasmid. So we've already talked about these, and I'm not going to spend time here going over that again, but I just want to call your attention to this figure right here. So you know that restriction endonucleases recognize a specific site, and when they see their site, they cut the DNA, usually both strands, and oftentimes they leave sticky ends like you see here. Now what we're going to need to do is whatever sticky ends are left over, when we cut our DNA of interest, the piece that we want to insert into the plasmid, we need to make sure that the ends of our piece of DNA are compatible in sequence. In other words, they're complementary in sequence to the sticky ends left behind on the plasmid. That way, when we take our piece of DNA of interest and try to insert it inside, the ends match and they can hydrogen bond with each other. All we would need to do is get an enzyme to connect the individual strands together to make that covalent linkage there to make it all one piece. The enzyme that does that is DNA ligase. It's the same enzyme that you saw during DNA replication that would ligate or connect together Okazaki fragments. So once you get your piece of DNA of interest, you isolate your DNA of interest, then you need to insert it into a cloning vector. And so that's basically like what we just said. that would involve cutting your piece of DNA of interest or making sure the ends of it are compatible with the ends that you have created on your plasmid by cutting it with a restriction endonuclease. If the ends match, then you can just add ligase, DNA ligase, to connect the ends together. Then you've created a plasmid that contains your DNA of interest. The next step, or the third step, would be to take your plasmid and put it into the organism that you've chosen. So my example is going to be bacteria or E. coli. So to get a piece of DNA into the E. coli cells, we would use the process of transformation, which you guys did when you guys transformed E. coli with a plasmid containing GFP. So you would perform that process, which involved making the cells competent, right? Incubating the competent cells with the plasmid and then putting them onto petri dishes that select for plasmids, or for cells, that got the plasmid and didn't let the cells that never received the plasmid, it doesn't let them grow, right? You would have to impose that selection using the selectable marker on the plasmid. So once you do the transformation, you'll get bacteria cells that contain your plasmid with your DNA of interest. Then all you have to do is take those bacteria cells, grow them in culture, put them into liquid media, liquid food, and grow them up overnight. And these cells, as they divide and replicate, they will have to replicate their chromosomal DNA and they will replicate the plasmid DNA. And so all the new cells that you make, they're going to be containing your plasmid DNA. So effectively, they've made copies for you, right? Any new cell should have a copy of your plasmid. And so that's what DNA cloning is about. You're using an organism to make many copies of your piece of DNA of interest. At that point, if you just if you want to then start collecting your copies, what you would have to do is then kill the bacteria cells, extract the DNA, extract and purify it, and then you can start to do whatever research or analysis that you wanted to do.
Another biotechnology application is when scientists want to express DNA, so make a product from the DNA information. Now, why you might want to do this? Perhaps you might want to make medicine. So, for example, the first human drug that was manufactured in this manner that we're going to talk about was human insulin. Before it was manufactured in this way, they used to have to get it from animals or cadavers in order to treat patients with, for example, type 1 diabetes. Or perhaps your goal is to purify a particular type of enzyme that you need in a different technique. Or, you just want to express a particular protein in a cell. And what you would need to do something like that, it would just basically be the same procedure we just finished talking about. But what would be different is instead of using a cloning vector, you're going to be using what's known as an expression vector. An expression vector has all the other elements that the cloning vector had, which was a selectable marker, an origin of replication, and then a multiple cloning site to insert your gene of interest. But what it has in addition to that is a promoter. And again, the promoter is important because it will allow you to actually turn on expression of your gene so that you can make a protein product. And so you definitely need that if you want to express your DNA. You need a promoter. This is the general workflow. Again, very similar to what we just discussed. You're going to need to isolate your gene or DNA piece of interest. You want to then insert it into a vector. And again, instead of using a cloning vector, the big difference is here, you're going to make sure you use an expression vector that has a promoter to control your gene. Then you take that vector with your gene and you transform it into an organism, usually a microbe like bacteria or yeast. And then you impose a selection to make sure that you're only growing the microbe containing your vector, your expression vector. And then you grow up the culture of that organism. You grow up your microbes, right? And as they replicate and grow, they're going to make your protein as long as you've turned on the promoter. And once you get a certain concentration of organisms, you can then lyse the cells, break open the cells, and extract and purify the protein. We're back to this figure where again you have your microbe, in this case they're showing bacteria, that contain your plasmid of interest. Now we're saying it's an expression vector, and so it's turning on the protein, or turning on the gene so that the protein can be created, and the protein's little purple dots. And after you extract the protein, you harvest it, you can use it for whatever purposes. Perhaps you're using it as a drug, right? It's helping to dissolve blood clots, or maybe you're purifying human growth hormone. So it really just depends on what this protein is and what your goals are.